So in this Hughes Views, I'm going to be covering fatty acid synthesis. This follows many of the concepts out of chapter 16, section 2 in your textbook. All right, so let's just think really big picture right now. When is it that your body is going to make fatty acids? And it has to deal with cellular respiration. So typically, if you've eaten too much food, right, you're going to store that extra food as energy for later in case times of starvation come around. So when you eat food, a lot of your body ends up funneling a lot of those metabolites into cellular respiration. So lots of the sugars go through glycolysis, and glycolysis ends up moving into the Krebs cycle or the citric acid cycle. A lot of different proteins enter either at some point in glycolysis, or they can also enter in the Krebs cycle at different locations. But so as nutrients are being processed by your body and running either through glycolysis or through the Krebs cycle, you're oxidizing those molecules. So you're getting rid of a lot of electrons. So you create these electron carriers, which is the cofactors we will talk about in class or have already talked about in class, of NADH2 and FADH2. And all these carriers, so as you stripped a bunch of electrons off of the food that you ate and you put them into these new molecules, those get fed into the electron transport chain and generate a whole lot of energy. So how does fatty acid synthesis relate to this? So the key part of where it actually relates is, sorry, let me grab the pointer, is right here, acetyl-CoA. All right, so if you eat way too much sugar and you're going through glycolysis and you make your 2 pyruvates at the end of glycolysis, and then pyruvate moves into the mitochondria, and then that gets converted into acetyl-CoA, you're going to have a lot of acetyl-CoA around. And so if there's too much acetyl-CoA that your body has made because you ate too much food and it couldn't process it, right? You didn't need all this energy. It's going to have this, just imagine the concentration of acetyl-CoA in your mitochondria just getting bigger and bigger and bigger and bigger. And it's like swamping out all the molecules. So there's too much acetyl-CoA. Your body is going to start saying, all right, there's too much of this stuff. Let's go turn this into fats, and we're going to store it long term. Like there was too much food that was eaten. Let's store it. So acetyl-CoA is the link between cellular respiration and then making fatty acids. So again, remember, big picture, you eat too much food, you consume too many calories, you are going to convert that into fat to store for a long time. And essentially that's all going to funnel through this one molecule, acetyl-CoA. And so this is the structure of acetyl-CoA. This whole part in black is the coenzyme A, cofactor. And then here, right here, is this tiny little acetyl group that's only 2 carbons. So here is acetyl-CoA. So this is what your body makes from glycolysis. And remember, if you had too much, then the concentration of acetyl-CoA gets too big. And then your body says, all right, we got to do something with this. So you're going to start funneling this excess acetyl-CoA into making fatty acids. So this, again, occurs in your fed state, like when you've eaten a lot. So you've got insulin pumping around. Insulin also, if you've watched the JJ Medicine video, insulin activates acetyl-CoA carboxylase, which adds a carboxylic acid moiety right over here and turns this into malonyl-CoA, which is the extender unit to add on 2 carbons onto a growing fatty acid chain. Okay, so, and we'll get there. Here's more detail coming up, don't worry. All right, so acetyl-CoA turns into something like palmitate. Palmitate is 16 carbons, right? And how does that happen? Again, it's going to occur in the fed state when you have insulin around, and if you consume too many calories, you're going to start switching that over into converting into fat for long-term energy storage. Here is the overall net chemical reaction. So it says acetyl-CoA plus 7 malonyl-CoAs. If you recall from the JJ Med video, that each malonyl-CoA was made by essentially adding a CO2 onto acetyl-CoA. So you can almost think of these 2 components in themselves as 8 acetyl-CoAs. And that'd be okay to think about if you asked me. So 8 acetyl-CoAs plus 14 NADPH plus some protons yields palmitate, this structure here. So it's 16 carbons. Remember, we're adding 2 carbon units on per time, so that should be 8 total rounds. Hence, you have 8 total acetyl-CoAs, right? 7 CO2s, those are coming off of this malonyl-CoA. I know, it seems a little redundant, like you took acetyl-CoA, you put a carboxylic acid on it to make malonyl-CoA, and then as you're adding and increasing your chain length of a fatty acid by 2 carbons, you're going to get rid of that CO2. It's just your body doing its thing. You're going to oxidize the NADPH to NADP+. You're going to free up 8 coenzyme A's, and those are coming from 1 from the acetyl-CoA and 7 from those extender units, the malonyl-CoAs. And then you're going to make some water. All right, so about the pathway of synthesizing and building up a fatty acid, it's pretty cool. There are 3 typical types of pathways that you'd find in your human body. You can have a linear metabolic pathway, and a great example of this that you're familiar with is glycolysis. You can have a cyclic pathway, and the one you should be familiar with at this point in your career is the citric acid cycle. So that is more of a cyclic pathway. And then lastly, the one that's really fun and really only occurs in very few cases in life is a spiral pathway, where you essentially have one single enzyme and the substrates just keep spiraling around onto different domains of that enzyme. And that's how you're generating this bigger and bigger and bigger molecule. So our fatty acid synthesis is an example of a spiral pathway. So again, and every time we go around the enzyme active sites, you're going to be adding 2 carbon units each time. Ding, 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 ding. So you go increase by 2s. So this is the structure of the enzyme that actually does the synthesis. It's called the fatty acid synthase. And it's a massive complex. It's huge. And it's a dimer. Not only that, it's a homodimer. So you can see here in this figure from your book, it's got the dimer axis here. Like I had said earlier, it's made up of many different domains. At least in this structure, 2 domains were not present, so they've kind of added them in here in this little colored like image, those little empty circles here. And so we're going to go through each of these domains, and I'm going to tell you the chemistry that kind of occurs at each domain. I might skip forward a slide though, and we'll look at the process of how we add 2 carbon units at a time, and then maybe go back. I think I'll try that first. We'll try that. You can always rewind the video for more clarification. Okay, so again, the spiral pathway, we're going to add 2 carbon units on per time. And then one thing that's early in your book, I believe it's in chapter 1 or 2, this fatty acid synthase, the fact that it has multiple domains that work in the reaction of building up a fatty acid is a great example of metabolite channeling. And that just means that the product of one reaction is immediately put into the essentially the active site of the next enzyme that needs to work on it. So you're channeling this metabolite through all these different domains within this one enzyme complex. Actually, it's just an enzyme dimer, but it's multi-domain, has many domains. All right, so we're going to come back to this slide. This is the one where it just tells you what each domain does. But let's look at kind of how the synthesis works here. So we can start, we're going to start here down at the bottom of this, and we're going to come up and work our way up here. All right, so remember in our reaction, the reaction is acetyl-CoA plus 7 malonyl-CoA. But so we're essentially going to react an acetyl-CoA with a malonyl-CoA. So this down here is kind of like the example of the acetyl-CoA. Forget that R group, like if you want to just think of it as acetyl-CoA, it would only be these 2 carbons only. But since it's the spiral pathway, this is a snapshot of kind of one round somewhere in between building up one of these big fatty acids. But so let's pretend that those 2 the carbon in the R group over here don't exist. So it's just like acetyl, and this would be CoA, not ACP. We won't worry about ACP right now. I just want us to focus really on the chemistry of what's going on. So we would have this acetyl-CoA group, and then here is our malonyl group. So it's that carboxylated, there's our CO2, onto an acetyl-CoA, or don't worry about ACP right now, we're just thinking about things in terms of CoA. All right, so we're going to take this acetyl group and we're going to condense it with this malonyl-CoA and release this carboxylic acid. As far as the mechanism goes, how that occurs is the carboxylic acid, the electrons come down to form a CO2 molecule, and then these electrons jump up on the carbon to give you a carb anion, which then that carb anion, very reactive, attacks this carbonyl. Electrons jump up, jump down, and are going to kick off that coenzyme A. And so what you'll see is you extend that your acetyl group down here by 2 carbon units here. And note that blue was our extender unit, that malonyl CoA, and that group is closest to the sulfur that we're always going to be building on. Okay, so in the process of that whole chemical reaction though, so remember, you decarboxylate here, these electrons jump up on the carbon, give you a carb anion. That carb anion attacks this carbon here, and electrons jump up, give you an oxyanion intermediate, and they jump back down to rebuild that ketone, and they break that CoA bond, this CS bond, this thioester bond. It's a very high energy bond, very, very favorable to break. And so you're left with this, which is a beta keto acid. So you have, here is, remember, nomenclature. This would be our first priority carbon right here. So that would be the alpha carbon, and then this is the beta carbon. So this is a beta keto. Also, beta is the 3rd carbon. So they say 3 keto. You can also say beta keto. Okay. So we have a beta keto, and if you think about the structure of a fatty acid in your brain, there's no alcohols or ketones anywhere, right? It's just completely reduced carbons. Right now, this carbon is fairly oxidized, so we need to reduce it. Remember, when we're reducing things, we're going to be adding electrons onto that thing to reduce it down. Okay, so then this is going to come into contact with 3 different domains within that big fatty acid synthase. The keto reductase domain, and that's going to reduce the beta keto down to a beta alcohol or a 3-hydroxyl. So and to do that, to reduce down, we have to add electrons, and those electrons are going to come from this NADPH cofactor. So we're going to put essentially 2 electrons in this hydrogen on this atom to reduce the ketone down to the alcohol. So that occurs in the keto reductase domain. Now we have a beta alcohol, and again, we need to essentially fully reduce this down so that it's only a hydrocarbon. So we still have this alcohol, we got to get rid of it. So the next domain is called the DH domain. That stands for the dehydratase. And so what we're going to do is we're going to remove a water molecule from the alpha and beta carbon. So here's our OH, and if we grab one of these H's, we can have our H2O molecule. So if we dehydrate and lose a water molecule, you will make a double bond. And it's going to be a trans double bond. So you make a trans double bond here. Notice, like, we started off with a ketone, then we reduced that to an alcohol, and now we've reduced that down to an ene, like a double bond here. All we have to do now is reduce it just a little bit further, add a couple more electrons to it and a hydrogen, and we can get the fully reduced CH2 functional group, methylene functional group there. And that occurs at the ER domain on that massive enzyme complex, and that stands for the enol reductase. So it reduces the double bond down to a single bond. And in order to do that, it requires the addition of electrons, and those electrons are going to come from the NADPH cofactor. So, all right, so that's how. So here we added these 2 carbon units onto our chain. And then when we did that, though, we formed this beta keto group. And then the part of the fatty acid synthesis now is you just need to reduce that down all the way to the methylene. So all hydrogen. So first it reduces to an alcohol, then to a double bond, and then to the single bond. And now this is the substrate for the next round. Again, that's spiraling on this enzyme. It's one enzyme doing this multiple rounds. It only goes around 8 to 9 rounds, if I'm doing my math right in my head. And then it cuts it off. It gets too big and it can't work with that substrate anymore. But so this becomes the starting material for the next round. So you take this molecule, right, and you're going to add another extender unit. So another malonyl ACP comes in. In fact, over the course of building up this palmitate, right, you're going to go through 7 rounds of extension where you're adding on, extending with these malonyl groups. So you extend with this malonyl group, the decarboxylation happens. It's called a decarboxylative Claisen-like condensation, if you want to get really technical. But you decarboxylate, make a carbon ion, and it attacks here. Electrons jump up, jump down, and kick off that ACP, and then there you go. Bob's your uncle, you got your beta ketone. And then the next three enzyme domains on that fatty acid synthase: reduce, dehydrate, and then reduce again. Okay, so let me jump back up a slide now. So that kind of talks about the cyclic nature of the spiraling and how that works. So let's talk about the different domains one by one and where they're located, because these are some of your learning objectives, is to be able to identify the different domains and then tell me what they do. Okay, so the first domain would be the KAS. It stands for keto-acyl ACP synthase. So KAS domain. And this is where that Claisen-like condensation reaction is going to occur. So this is where the extension actually occurs at the KAS domain. This creates your beta keto functional group on your growing fatty acid chain. So that's here, right here on this dimer interface. So that's the KAS domain. The next domain is the malonyl acetyl transferase. So that's the MAT domain. And this is what accepts either the very first starting acetyl-CoA, or if we're talking about palmitate, the 7-malonyl-CoA extender unit. So all those first have to come on here to this domain, the MAT domain, hence malonyl/acetyl transferase. But mainly in the grand scheme of things, right, it's like 1 acetyl and 7 malonyl groups. So I just said accepts the malonyl extender, although it does take the very first one too. Okay, so essentially think, okay, so we have our extender unit, we had our previous smaller chain that's sitting on the KAS, and then we're going to condense those together at the KAS, and then next up is the processing at these three domains over here, the KRDHER. So the KR domain, which is located out on this outer side, it's like the KR is kind of out here, the DH is right here, and the ER is this one over here. So at the keto reductase domain, the KR domain, that's where we're going to reduce the beta carbonyl down to a beta alcohol, and that requires the use of the NADPH cofactor. The next domain that this will stop off to is the dehydratase domain, the DH domain, and that's where we're going to remove a water molecule from the alpha and beta carbons and be left with a double bond. Then we have the ER domain, the enol reductase domain. That reduces the double bond to a single bond and is going to require the input of electrons to do that reduction. Remember, oil rig, oxidation is losing, reduction is gaining electrons. So we have to gain electrons. And that comes from the NADPH cofactor. Okay, so then the last couple to talk about are, so that's the main players of the game. But now maybe the question is, how do these substrates move around all these different domains? Is maybe what you might be thinking. And that's where this ACP domain comes into play. This is an acyl carrier protein domain. And on that one slide we just talked about, you saw ACP a lot. And I was like, pretend that doesn't say ACP right now. Just pretend it says coenzyme A for all intensive purposes. So the ACP is what shuttles the growing fatty acid strand around all these different domains. It is a small triple helix protein that has a prosthetic group, so it's covalently linked. It's essentially the long arm of the coenzyme A. So it ends with that thiol, the SH, and so that's what you're going to move around all this growing fatty acid chain on, is this ACP. And on the next slide, I'll show you like the order in which the ACP moves things around. And lastly, you got to get this chain off this enzyme at some point. And so that occurs at the thioesterase domain. And this just simply uses a water molecule to hydrolyze the growing fatty acid chain off of the acyl carrier protein. So we just use a simple water molecule to cut off, essentially break that thioester bond, and you're left with your fatty acid with the carboxylic acid moiety, and then your the coenzyme A prosthetic group that's on this acyl carrier protein takes the other hydrogen from the water, H2O. Okay, so ACP moving stuff around. So let's talk overall about how the cyclic kind of reaction works. So remember, the MAT domain could accept a malonyl or the acetyl-CoA as an extender unit. So this seems kind of weird, but I'm going to walk you through like maybe 2, maybe 3 cycles. Let's walk through 3 rounds of rowing fatty acid. Okay. All right. So on the very first round, nothing's happened yet. All of these domains, so we have the KS, that's where we're going to condense and actually make the carbon-carbon bond. You have the KR, we're going to reduce. You have the DH, we're going to remove water. You have the ER, we're going to reduce again to a single bond. And you keep cycling through all these things, and then eventually you cut off with a water molecule by the thioesterase domain. Okay, so the ACP is what's carrying all this stuff around. Okay, so very first thing that occurs is the MAT domain. Again, everything's empty right now. Nothing has anything bound on it yet. So a free acetyl-CoA will come and bind to the active site serine covalently. It'll covalently link to the active site serine of the MAT domain. The acyl carrier protein with its long prosthetic pantethene arm comes in and picks up that acetate unit right here on its sulfur. So now you can imagine this could have this structure, but instead of saying S-CoA for coenzyme A, it would say S-ACP, because now it's on the acyl carrier protein. So we have the acetate unit on the acyl carrier protein. This then moves over to the ketosynthase and transfers this 2-carbon acetate unit onto an active site serine of the ketosynthase. So now this 2-carbon unit is covalently linked on the KS, and the acyl carrier protein is free, is carrying nothing again. It just has its like long arm. It's empty now. So that's how acetyl-CoA is primed on this. So it comes first into the MAT domain. ACP comes and picks it up and moves it over to the ketosynthase domain and drops it off like as a covalently attached acetate unit. And the ACP is free again. So again, we're on first round of condensation. Next, a malonyl coenzyme A will come and bind to the active site serine of this MAT domain. And the acyl carrier protein will come and pick up this malonyl group. Okay, so instead of this saying S coenzyme A, pretend now it says S-ACP, but it has this structure. Then pick up that malonyl group, and it's going to go over to the ketosynthase domain where this is already waiting, and it's going to perform that Claisen-like condensation. So where it decarboxylates, you get the carbanion, and this attacks on the ketosynthase domain. These electrons jump up, jump down, and essentially kick off the ketosynthase domain itself, right? And then you've extended your carbon chain by 2 units. So now you have 4 total carbons in your fatty acid chain. And the beta keto. So the ACP now moves over to the KR, which reduces the beta keto to a beta alcohol. Then to the DH, which reduces the alcohol down to a double bond by removing a water. And then to the enol reductase domain, which reduces the double bond to a single bond. And now you have a 4-carbon chain fully reduced like fatty acid tail, still covalently bound to this acyl carrier protein though. So the acyl carrier protein is going to now skip over and drop that off at the ketosynthase and like covalently link that onto the ketosynthase domain. So you have a 4-carbon fatty acid now linked up to the ketosynthase and a free acyl carrier protein. And now that acyl carrier protein is going to go pick up the next malonyl extender unit that's waiting for it at that MAT domain. So the ACP picks that up. You're going to see a covalent switch of these functional groups, like this malonyl group, onto the arm of the acyl carrier protein. And now it moves over to the ketosynthase, where you're going to see a Claisen-like condensation reaction. And you're going to take the 4 carbons now and turn it into 6 carbons. So now you have a 6-carbon chain and the beta keto on carbon 3. And now you just go through the process. KR reduces the carbonyl to an alcohol. DH removes the water molecule, creates a double bond. ER reduces the double bond to a single bond. And then the ACP drops that 6-carbon fully reduced chain off at the ketosynthase. And now the ACP arm is free. So where does it go? It goes to the MAT domain to pick up another malonyl group that's waiting for it, right? Look, we have 1 acetyl group we started with, and now we have to add 7 malonyl groups. So this this whole thing is going to occur 7 total times with the malonyl groups, right? So on and so forth. So you keep doing that iteratively, cyclically, well, not cyclically, in a spiraling where we're growing 2 carbons every time we're like cycling through, and eventually it will get hydrolyzed off at the thioesterase domain. And I have a slide later that explains when that happens. So, okay. So again, here's that reaction. Maybe we can run through it real quick. So at the keto synthase, you'll have this molecule. This probably should, for more clarity, and have this like S, maybe say like O, SER or KS domain, right? Because this is going to be essentially left off and be covalently linked on to that KAS domain. But here comes our ACP. So that free ACP picked up the extender unit here and comes to the KAS domain where this reaction occurs, and you extend the fatty acid chain on the acyl carrier protein. So it just like takes, it's going to be added onto the acyl carrier protein. So now you've added 2 carbons to it. Then the ACP moves to the KR and it gets reduced. The ACP moves to the DH domain, it gets dehydrated. And then the ACP moves to the ER domain where it gets reduced again. And then this gets dropped off at the KS domain where it's covalently like moved over. Okay. Woo. So spiral pathway, multiple domains in a spiral-like fashion can essentially take, I'm stretching here a little bit, 8 acetyl coenzyme and turn it into 1 palmitate or palmitic acid, which would be 16 total carbons. Remember, each one of these is bringing 2 carbon units at a time. And I say 8 acetyl-CoAs because it's 1 acetyl-CoA and then 7 malonyl-CoAs. But remember, to make a malonyl-CoA, you take the acetyl-CoA and the ACC enzyme, the acetyl-CoA carboxylase, adds a CO2 molecule right here at the end of this. So you get your malonyl group that way. Okay, so how long will this happen? So chain termination. Chain is going to elongate until it's between 16 to 18 carbons in length. That's between palmitidic acid and stearic acid, right? Stearic is 18, palmitidic is 16. Then it will be transferred onto the active site serine of the thioesterase domain and hydrolyzed off with a water molecule. So why does it only create something that's 16 to 18 carbons in length? We definitely need fatty acids that are longer than that for our biochemistry, but that's just as far as we can go. This has to deal solely with substrate specificity in mammals. Bacteria are slightly different, and fungi are different, and plants are slightly different, but in mammals, our fatty acid synthase stops between 16 and 18 carbons in length. What does substrate specificity mean? Well, it means a couple things. So the TE active site can fit only something that has a minimum of 16 carbons in it. So if the chain is less than 16 carbons, if it moves in, like the ACP accidentally goes over and checks out, hey, is this ready to go, and puts it in the TE active site, but it's only 8 carbons, it's not long enough, and the TE can't grasp onto it and put it in the right position to actually get that catalysis to occur. So that's what substrate specificity means. It's like the substrate has to be very specific for the enzyme active site. So the TE active site fits a minimum of 16 carbons, and then the keto synthase can't accept anything greater than 18 carbons. So if you have the 18 carbon chain and you're trying to add 2 more onto that, the KS can't accept anything that big. So that's what limits your this chain length, is both of those substrate specificities. Okay, so how do we make fatty acids that are longer? So do not fear, we have more enzymes to help do that. So let's say we only made a palmitate, so 16-0, our fatty acid, and we can elongate that, or we can also add double bonds. So remember, like making some cis double bonds to make those types of cis fatty acids. And the first thing you have to do is you have to activate that palmitate to palmitoyl-CoA. So you take free CoA, and this is our palmitidic acid right here, and you're going to essentially couple that to the hydrolysis of ATP to give you an acyl CoA. So think of like the long palmitate 16 carbon chain on this coenzyme A. Once you made that, so you've charged it up with the coenzyme A, this now becomes a substrate for different enzymes called elongases and desaturases. So elongases are kind of like the KAS domain, except for they can accept something that's bigger than 18 carbons. So it is essentially that same type of chemistry occurring, but it's just not occurring on our little spiral enzyme anymore. It's on a separate enzyme called an elongase, where you'll add 2 carbon units and then you go through another round of KRDHER processing, but those aren't the same as the ones that are on our 1 fatty acid synthase. They're different enzymes that do that. So the elongase, which doesn't use an acyl carrier protein, will take the malonyl CoA and add those 2 carbon units, and then you'll do the reduction, dehydration, and reduction again to give you those additional 2 carbon unit extensions. So now instead of, well, this one we started with stearic acid, 18 carbons, right? Now we have 20 total carbons here. And then what about making double bonds in our fatty acids? So not all fatty acids are saturated. We know that there are double bonds. We have unsaturated fatty acids. So a couple key differences maybe between bacteria and eukaryotes. So bacteria can actually make the double bonds while they're building up their fatty acid chains. Their KAS domain can recognize the kinks that would occur in those unsaturated chains. And so that's really cool. We can't. That sucks. We can't do that. So the eukaryotic fatty acid synthase cannot make double bonds. We have to do that after the fact. So we only end up making long saturated fatty acids, and then we can go through and tailor it and edit it and change it. And that's because of substrate specificity. Our KAS active site can't allow for any types of long chains that have these kinks in them that are, geometrically constraining that the free rotation of the molecule itself. Okay, so what would that look like? So, and what are these enzymes called? So enzymes that create double bonds are called desaturases. So they desaturate, they remove a water molecule. And so you can see that requires oxygen and electrons from NADH, so the NADH cofactor. And I am going to save you and spare you the mechanism behind it. But essentially, you can take a double bond. You can take, a single bond and turn it into a double bond. And typically, the enzymes that desaturate have this type of a nomenclature where it's the delta with the superscripted number, and that tells you the carbon at the lowest carbon and the double bond at which they're going to make that double bond at. So this is a delta 6 desaturase. So here's our carbon 1 with the carbonyl. So 1, 2, 3, 4, 5, 6. So it's going to take away or make a double bond between carbon 6 and 7. So I sometimes might ask you, like, what would the delta 9 desaturase do to that fatty acid? And so you would have to draw a product showing that it made a double bond between carbons 9 and 10, right? Because it's a delta 9 desaturase. So that's the lower number in the fatty acid chain. All right. So what are some takeaways of this lecture? It's a very long lecture, but it's really, really cool, fascinating stuff, especially because there's not that often you get to see this spiral pathway. So one enzyme just iteratively doing the same, same chemistries again and again to build something up. Okay, so you should be able to sketch and label the domain locations on the mammalian fatty acid synthase. So that's that one structure in your book that looks like a pymol structure with the alpha helices and the beta sheet. So you should be able to just label the general location of the KAS, the MAT, the KR, the DH, and the ER domain. Remember, the ACP and the TE weren't solved, so that's fine. You don't have to label those ones. You need to be able to provide a description of the reaction each domain catalyzes and include all necessary cofactors. So that's for the KAS, the KR, the DH, the ER, the ACP, which doesn't really catalyze a single reaction, right? That's more the carrier that carries it around everywhere, but it does have a prosthetic group that is derived from a cofactor. So you should know what that is. And then lastly, the thioesterase domain. You should be able to sketch the structure of a growing fatty acid chain after each reaction. So what would the product of the KAS reaction look like? What would it look like after the KR worked on it? You know, what's the before and after of a DH domain? What's the before and after of the ER domain? So I would like to see you processing that, making that beta keto and then processing that beta keto. Summarize major products and energy requirements to synthesize one molecule of stearic acid, C18. Note there is an equation, especially the one I think we went over a couple times in here, and that has to deal with palmitate, so that's a 16. So you need to adjust that equation accordingly to account for one additional extension reaction. So to go from 16 to 18. So you might have to do a little math and a little thinking, but Google is also your friend. But try thinking it first and then check yourself. And lastly, compare and contrast the elongation and desaturation reactions that occur between bacteria and eukaryotes during fatty acid synthesis. I only talked a little bit about one or two differences, but your book lists a lot more differences that occur. So make yourself a nice little, Venn diagram comparing and contrasting bacteria elongation versus eukaryotic elongation, and then bacteria desaturation versus eukaryotic desaturation. And that completes, the lecture.